NDVI estimates in cotton performance trials and compare the efficacy of different experimental designs and statistical analyses at reducing experimental error. So I'm going to start by giving an overview of high-throughput phenotyping research efforts in cotton. My focus is going to be limited to ground-based or proximal measures, but it's worth noting that there's been a lot of research done and a lot of research currently being conducted using aerial platforms as well. So in 2014, a group in Maricopa, Arizona, including Pedro Andrade Sanchez at the University of Arizona, and then Mike Gore, our host today, while he was with the ARS, reported the first ground-based high-throughput phenotyping platform, which deployed a GPS receiver and multiple sensors mounted on a high-clearance tractor that measured canopy reflectance, temperature, and height on 4 rows simultaneously. So this platform was used to evaluate in-season canopy traits of 25 Pima cotton cultivars under both well-watered and water-limited conditions. And so what they saw was a strong agreement between manual phenotypic measures and aerial imagery, and they were also able to detect significant differences between cultivars, as well as a significant three-way interaction between cultivar, irrigation regime, and time of day. So then in 2015, a group out of Lubbock, Texas, including Glenn Ritchie, used a similar phenotyping platform to evaluate seasonal growth and plant structure of 4 upland cultivars managed under 10 different irrigation levels. So using this system, they were able to detect significant differences for these canopy traits between irrigation levels and cultivars. And also of note is that they observed the relationship between these canopy traits and lint yield were the strongest during flowering. So then in 2016, the group out of Maricopa, including Duke Poly, who's now at the University of Arizona, used their platform to evaluate stress-responsive traits, specifically heat and drought tolerance in a recombinant inbred line population in both well-watered and water-limited trials. And so from these data, they identified temporal patterns of QTL expression for these canopy traits. And similar to those observations in Texas, they observed that the strongest relationship between these canopy traits and lint yield occurred during a time period of flowering. So in Florence, we have a similar ground-based platform to those that were deployed in Arizona and Texas, where we can measure canopy reflectance, temperature, and height on 4 rows simultaneously using high-clearance tractor. However, today I'm just going to focus on our canopy reflectance measures, specifically Normalized Difference Vegetation Index, or NDVI. So generally on a broad scale, our breeding program is exploring the use of high-throughput phenotyping to identify and characterize drought-tolerant germplasm, as well as select for yield potential in early generation material. And some of the important factors that we have to consider that are maybe less important in Arizona and Texas, incorporating this into our breeding program, is that the climate and the soils in the southeastern U.S. are very highly variable. So for example, in the early 90s, the NRCS conducted an intensive soil survey at the Clemson Experiment Station located in Florence. And you can see from this map that up to 10 different soil types can be found in a single field. So previous studies in cotton have demonstrated that field heterogeneity like this can reduce the reliability and precision of lint yield estimates. And it's also well known that high throughput phenotyping traits like canopy reflectance or NDVI are influenced by environmental factors like soil fertility and moisture availability. And so one of the first questions we wanted to address as we started to incorporate this technology into the breeding program was how environmental variability affects these HTP measures. So the objectives of this work were to evaluate the efficacy of different experimental designs and spatial analyses in the estimation of genotype effects for NDVI, and also to measure the impact of different experimental and statistical approaches on selection for both NDVI per se and NDVI as a proxy for lint yield. So for this study, NDVI was measured on 3 performance trials in 2014 and 15 in Florence. Each trial had 288 entries each, arranged in an alpha lattice design with 2 replicates. Additionally, alternating rows of a common cultivar, Phytogen 499, were superimposed on the incomplete block design, such that every 2 rows of Phytogen 499 bordered every 2 experimental rows. And so this alternating check design was used really to evaluate the efficiency and practicality of planting a common genotype to adjust for field heterogeneity. So we measured NDVI when approximately 50% of the genotypes had reached first bloom. And this translated to around 45 to 60 days after planting. And unfortunately, we were only able to measure lint yield on trial 1 due to a historic rainfall event in the fall of 2015 that ultimately destroyed the trials planted, at least the 2 and 3 trials planted in 2015. So to process our canopy reflectance or NDVI measures, we created Polygon boundaries, seen in gray, based on known field and plot dimensions, as well as the quantities of the georeferenced NDVI measures. So our GPS receiver has sub-meter, but not RTK level accuracy. And so at this point in our process, the data isn't split into individual rows. Instead, these polygons represent tractor paths, so a combined four rows. We then clip and append the polygon attributes to the point data. And so each polygon has a unique ID, a range or column number, as well as the direction the phenotyping system was traveling. And so with this data, we're able to assign plot IDs based on the polygon attributes, as well as the sensor number associated with the point data, and ultimately calculate our plot means. And so NDVI, as well as lint yield data for trial 1, were analyzed separately using 5 different mixed models. The randomized complete block model was used as the baseline, where our genotype effects were treated as fixed and our complete block effects were treated as random. So the second model included the addition of a random incomplete block effect, corresponding with the alpha lattice experimental design. And then thirdly, the efficiency of a row column incomplete block design was investigated using a post-blocking exercise where random row and column effects were added to the baseline RCB and analyzed as if they were true incomplete blocks. And so this method of post-blocking is not recommended in routine analyses of plant breeding trials, but it is frequently used to investigate the efficiency of alternative experimental designs. And so our fourth model included nearest neighbor adjustment for field heterogeneity based on the measurements of the alternating check rows of phytogen 499. So the average of the residuals from the east and west bordering check rows of each experimental plot was added to the baseline RCB as a covariate. So these first four models assume independence among plot errors, and research suggests this is rarely valid for RCB designs when complete blocks have greater than 10 genotypes. So we also investigated the efficiency of spatially correlated errors models, which model the covariance between plot errors as a function of spatial distance. And so in our case, the spatial distance between plots was coded as the UTM coordinates for the center of each plot. And so for these spatial models, we evaluated several common covariance functions, including the power, Gaussian, and spherical functions, which model spatial dependence or autocorrelation independent of direction. And then additionally, we evaluated the separable first-order autoregressive function, often referred to as AR1 by AR1. And this function models spatial dependence in both the row and column directions separately. And so these spatial models were also evaluated with and without a nugget effect. So among these 5 models, we compared the relative quality of the nested models, which include all of them except the nearest neighbor adjusted, based on log likelihood ratio tests and AIC values. All 4 alternative models were also compared to the RCB in terms of precision or discriminatory power between genotypes. And we did this by calculating relative efficiency, which we measured as the ratio of the average standard error between pairwise genotypic differences between the RCB and the alternative model multiplied by 100. And then lastly, we compared the percent agreement between genotype effect estimates from different models, looking at both the Spearman correlation coefficients and the Chekhanovsky coefficients, where A represents the number of lines selected by both models, and C represents the number of lines selected by the baseline RCB only. And so we looked at this percent agreement between genotype rankings at several different selection intensities between 5 and 50. So this figure shows the distribution of NDVI at early bloom for the 3 trials. Our plot level measures ranged from 0.1 to 0.85, which is close to full canopy closure in cotton. And the mean NDVI for each trial was between 6 and 7, or 0.6 and 0.7, excuse me. So we investigated the presence of field heterogeneity or spatial autocorrelation by plotting the 2-row average NDVI of the alternating check rows of 499, and that's the figure you see on the left. Stronger spatial trends were apparent in 2015. And I should also note that the complete blocks were arranged in the column direction. So from these figures, it's pretty apparent that the RCB design is not optimal for reducing within block variance, particularly in trials 2 and 3 in 2015. So additionally, to investigate the influence of soil type on field heterogeneity or spatial trends in NDVI. We superimposed the soil maps onto the plot data. However, we didn't see a strong correlation between soil type and NDVI, perhaps with the exception of this Nabokos sandy loam in the top right corner of trial 3. So considering the trials were grown under rain-fed conditions, the spatial trends that we do see in NDVI may have alternatively been influenced by variation in soil moisture availability due to field topography or lateral water movement. And so we also looked at spatial dependence by plotting semi-variograms based on the RCV model fit residuals. So the positive relationship we see here between semi-variance and distance suggests that spatial dependence was present in all 3 trials. So then looking at the model comparison, the alpha lattice, row column, and spatial models were superior to the RCB in terms of log likelihood ratio tests and AIC values. And then all 4 models, including nearest neighbor adjusted model, improved the precision of genotype effect estimates as indicated by the relative efficiencies. With the one exception here of the nearest neighbor adjusted model in trial 1. So ultimately, the row column model provided the best improvement in precision compared to the RCB, resulting in a 20 to 66% reduction in the average standard error of pairwise genotypic differences. And I should also note that although genotype effects were highly significant in 2014, we weren't able to detect significant differences in NDVI between genotypes in 2015, with the exception of trial 3 when analyzed with the row column model. And so this table shows the comparison between the genotype effect estimates across models. First, on the left, shows the correlation coefficients between NDVI estimates. And one thing to note here is that the correlations between NDVI estimates from different models were generally lower in trials 2 and 3, where more extensive spatial trends were present. So among these 2 trials in 2015, correlations between NDVI estimates were greatest between the spatial and alpha lattice models, shown in green, and they were lowest looking at the comparison between the row column model and the RCB and nearest neighbor adjusted models. So on the right-hand side of this table, we have the percent agreement between the RCB and alternative models for genotypes ranked in the top 50%, or top 5 to 50%. And the percent agreement between those estimates of genotypes ranked in the top 50% ranged from 69 to 97% across the 4 alternative models. However, as selection intensity increased to 5 and 10%, the agreement between models decreased. So for example, in trials 2 and 3, the percent agreement between these alternative models and the estimates from the RCB never exceeded 53%. And so next, looking at the relationship between NDVI and lint yield, we observed a moderate positive relationship with R-squared values ranging from 23 to 27%. And despite those considerable rank changes in NDVI estimates we saw in the previous slide, similar proportions of the top yielding genotypes were selected based on NDVI independent of the statistical model. So these results, in combination with previous findings in cotton, suggest that NDVI measurements during flowering might be most useful for eliminating genotypes with low yield potential. And this is instead of selecting, say, the top 5 or 10%. And so to summarize, I believe these findings really highlight the importance of using appropriate experimental designs and statistical analyses to reduce experimental error in canopy reflectance measures, particularly in the presence of field heterogeneity or environmental variability. And I think these results likely extend to other high throughput phenotyping canopy traits. Our results also suggest that experimental designs, which account for variability in 2 directions, such as the row column design, might be preferable to the RCB or even alpha lattice designs, both of which are the most commonly used experimental designs in cotton performance trials across the U.S. And so these data also demonstrate that planting a common genotype to adjust for field heterogeneity is not likely the most efficient use of resources, at least for replicated trials. And lastly, the results also show that field heterogeneity can have a significant impact on selection decisions for NDVI per se. However, the impacts of environmental variation on the relationship between NDVI and lint yield warrants further study. All right, and so with that, I'd like to acknowledge my co-authors, including Todd Campbell, Phil Bauer, and Ken Stone, that also work at the USDA station in Florence, as well as support from Ernie Strickland and Kendrell Wincate, and financial support from Cotton Incorporated. And I would also like to thank the reviewers on this paper for their constructive feedback and making this a strong paper. And with that, I think I have plenty of time for questions.
All right. Thank you, Dr. Hugie, for the exciting seminar on incorporating experimental design and spatial analyses to help improve the precision of your NDVI estimates in the upland cotton field trials. This is obviously a very important area of research as it applies to plant breeding. And we would like to open it up now for the next 15 or so. Maybe, actually, we have a half an hour for a few more questions, which you can either write in your chat section directed to my name is Mike Gore, or to all, and I can also read them, or you can unmute by turning down your volume. While you're preparing your questions, I will make a brief pitch here for the Plant Phenome Journal. It's our new journal. That's why we're having this series of presentations to help promote this as a tri-society journal dedicated to advancing these issues, such as we just heard about from Kari. And the TPPG is accepting articles that push the frontiers of plant phenotyping, and it's helping to catalyze this new area. And I guess I will open it up. And one question I have for you, Kari, is you found that the soil maps didn't help improve very well, but I was just curious if you had maybe more detailed soil maps that, in terms of higher precision in the sampling, either in subplots, if that may change the actual finding when then you're including that maybe as a covariate in some of your findings.
Okay, yeah, thank you. I think as we're able to characterize the environment that during growth and development a little better, I think definitely that we'll see a higher correlation between, say in our case, NDVI and that's the spatial trends. Particularly in this case, I think if we were able to measure water holding capacity or soil moisture, but those soil maps were all we had at the time.
Right.
So we thought it would be interesting to overlay them and see what kind of trends we saw.
I don't think I've received any questions yet. Is there anyone online that wants to ask a question? If you unmute, or you can send it to me and I can read it, either way. Well, while we're waiting for that, I'll ask a question. So, Kari, I'm just curious what your thoughts are in the value of phenotyping like early in the season prior to flowering, because I think most of your phenotyping, and it makes total sense for this work, was I think at flowering and after. So I was just curious if there's other value, or if there's other phenotypes that maybe you think could be targeted like early in the season. And, of course, there's some of the challenges with the canopy hasn't closed fully yet, but I'm just curious if you thought about some of those areas or some of those trait questions.
Yeah, so we have thought about that. We typically look to use canopy temperature later in the season after there's been full canopy closure, but we have started to look and are starting to incorporate more early season measurements of reflectance prior to bloom to track growth and vigor during that early pre-squaring developmental period in cotton. And so some of the hypotheses we have, looking at maybe improved insect resistance, the faster it can grow out of the cotyledon stage. That might be an example. Yeah,
that's a really intriguing phenotype. And is there certain pests that you would want to breed for if you were to use that as a phenotype?
And so in the Carolinas, an early season vigor would be useful for thrips resistance, or at least that would be a hypothesis that we would like to test.
Sure, that sounds really good. So I haven't received, oh wait, I did just get a question, hold on, it just vanished though, where'd it go? In the chat, here we go. Okay. This is, I think, a question from Seth Murray. Seth, do you want to mute or do you just want me to read it? I can read it, it's totally fine. Okay, so the question from Seth is, if I understand correctly, you said row column was best. Why might that be? Was it due to furrow irrigation?
So I can only hypothesize as to why the row column provided so much additional precision. We don't furrow irrigate. All three trials were managed under rain-fed conditions. However, I don't know if you can still see my screen, but particularly in trials 2 and 3, in our alpha lattice design, direction of our blocks was in the row direction, whereas I think a greater amount of variability was seen in the column direction. And so by adding that two-directional blocking effect, I think added that additional precision.
Okay.
Hope that answers your question.
I think this may be a follow-up question from Seth. What about the fertilizer rig? Sometimes some rows get put out more. I guess he's referring to when the field is being fertilized. Yeah.
So the spatial trends can be due to management differences in management practices, as well as environmental factors. And so in this case, I'll keep this figure up. The plots would have been fertilized going the way we're looking at them, north to south. So according to the check rows, don't see much systematic variation that would be indicative of management practices.
Okay. From Francisco, great presentation, Kari. Have you thought about applying any kriging procedure to account for the spatial heterogeneity of the soil in the trials?
So we did think about it, and in the development of this manuscript, we decided not to make it about or make it focused on identifying the best like statistical procedure after the fact, and so we decided to limit it to some of the more common ones. However, kriging, or more recently, as Mike had suggested during the review process, some spline regression methods have shown some promise. And whereas the spatial models we consider, we kind of have to do in a sequential order to find the best covariance function, some of those methods simplify that process.
Okay. I'll see if there's a reply from Francisco, any follow-up, but I can go also. This is a question from Katie. How do you automatically get rid of outlier data points? If a spot in the field has poor germination or a spot has weed pressure, it will affect the NDVI values.
Yep. So the two approaches we take kind of depend on what we're looking to do with the data. We use values from the alleyways, so the soil properties sometimes, and create a threshold. So say if a measurement is within a standard deviation of the mean reflectance or temperature of an alley or soil measurements, we'll use that to detect and remove outliers. And we'll also use an iterative process in SAS, just fitting those point data to a mixed effect model and eliminating them with looking at the our studentized errors.
Yep, that makes sense. This is a question from Jared Crane. What software was used for the analysis, especially Model 5?
So we use SAS for all the analysis. Yeah. So all those covariance structures can be specified using proc mixed in SAS.
Okay. So I guess we've been going for about almost 10 minutes with questions. Perhaps we can maybe go for 5 more. Are there any more questions? You can either unmute your microphone and ask, or you can send me or to the whole group as a chat. While we're waiting for maybe any additional questions? Thank you, Kari, for putting that question slide up. Nice. So I guess my question for you, and perhaps this also ties in with like Todd Campbell's breeding program, is where you think the future of, say, the high throughput plant phenotyping research is for this cotton breeding program, and what type of impacts do you think it'll have on improving, say, the productivity of cotton over like 5 to 10 years out?
Okay, so my answer is probably twofold to that.
Sure.
So we're looking at it for two different purposes. I don't know if you can fully separate them, but first to characterize drought tolerance or drought responsive traits that don't necessarily correlate with lint yield. So just being able to collect measurements of on a high throughput scale of plant growth and development through the season is a really valuable for us, because one of the main objectives of Todd's breeding program is to identify climate resilient or drought adaptive germplasm. And then kind of the second part of that is on more applied side to the breeding program in terms of early generation selection for yield potential. And so there's a moderate correlation we've seen with NDVI and lint yield, and the same really holds for canopy temperature from what we've seen. And so we're looking at it to maybe expand early generation nurseries and be able to focus, eliminate the ones, genotypes or germplasm with low yield potential, and focus our efforts on phenotyping the remaining. So really increasing our numbers game. And that's how we started asking this question, was as our field sizes get bigger, do we need to change our experimental design or our, analytical approaches?
That makes very good sense. Thank you for that very comprehensive question. That definitely provides some good insights on where you see the high throughput phenotyping work going forward, but perhaps more importantly, how it's being used in an applied aspect, to turn that engine of evolution faster in plant breeding programs. So yeah, this is really good. Well, I'll give one more opportunity for anyone to ask a question. You can either unmute your mic or send it to me over chat. Well, I don't see any more questions coming in, so
thank you.
Kari, thank you so much for your time, and thank you for all that joined us. And if you have like a further question that maybe pops into your head, please feel free to follow up with Kari. Do you maybe want to provide your email address, Kari, in case anyone wants to follow up with you?
Yes. If you have any further interest or questions, my email address is kari.hugie@ars.usda.gov.
All right. Thank you so much, everyone, and I'll see you all in 1 month. All right. Thank you.